Hey y'all, it's Lance, and we are going to be going over the types of muscle today. So as we can see, there's going to be three different types of muscle that we're going to go over. We're going to have smooth muscle, cardiac muscle, and skeletal muscle. So let's go ahead and get into this and start learning about these muscles. All right, so first off, a muscle is going to turn chemical energy, and that's going to turn into mechanical energy with skeletal muscles. And so that does that when a muscle contracts across a joint. And so that chemical energy that is stored in that muscle is then going to be converted into that mechanical energy, and that's how we're going to be able to move. And then also with the contraction, what exactly is a contraction? So a contraction is going to be moving one bone closer to another bone by pulling across that joint. And so we have different types of movement. We're going to have flexion and extension. And so a really common example with this is going to be with the biceps brachii versus the triceps brachii. And so when we have a contraction, such as with the biceps brachii, it's going to pull the radius and the ulna closer to the humerus. And so that's going to be the action of the biceps brachii is to flex that elbow joint like so. But it doesn't extend the elbow joint as well. And so we need a different muscle group to do the opposing action. And so that's where the triceps brachii comes in for the extension. And so each muscle group is going to be in charge of either a flexion or extension. One muscle group isn't going to do both the flexion and the extension. It's going to be one group for each action. So it's like a little preview about like skeletal muscle and all that good jazz. So let's get going into the different types of muscle now and exactly what they look like and some different characteristics of each muscle. Alrighty, so let's go ahead and talk about smooth muscle. So smooth muscle, as we can see right here, they're going to be kind of short. They're going to be tapered at the ends, as we can see right here and right here. And let's talk about these different characteristics that we're going to be seeing. So let's take a look at this picture again. And how many nuclei are we seeing in there? We are only seeing that one nuclei that we see right here, right there in the center. They're not always going to be right there in the center. It's just for this image. So each of these smooth muscle cells are going to have one nuclei that we can see in there. And as we can see, compared to like cardiac muscle and skeletal muscle, they don't have those striations. And so this is going to be a non-striated muscle cell. So non-striated. Alrighty. And then the control. So when we think of smooth muscle, we think of like the muscles of like the organs, the airways, and the blood vessels. And so those are the locations that we're going to find these smooth muscle cells. And when you think about that, our organs, we don't really consciously think about moving food down our esophagus. It's that smooth muscle that does it for us. And so the control is going to be involuntary control. So involuntary right there. And basically, that is good because I personally wouldn't want to think about swallowing all of my food all the time and just like thinking about getting that down to my digestive system and then thinking about having to digest that. It just does it automatically because our bodies are just amazing like that. And so that's going to be the control for the smooth muscle. So those main characteristics that we see are going to be the nuclei, or we're going to have one nuclei in each smooth muscle cell. They are going to be non-striated, and the control is going to be involuntary control. Alrighty, and just remember those locations are going to be in the organs, our airways, as well as our blood vessels. So that's a little overview for these smooth muscles. Alright, so now let's go ahead and talk about cardiac muscle. So we're going to go over these same characteristics. And so if we were to look at this one, it looks a little bit different. Actually, I would say it looks a lot different than the smooth muscle cells. So we can see right here, if we look at these individual cells, so this would be a cell, this would be a cell, and this would be a cell. This whole thing is more than one cell for this image. So we can see that in these cells, there is just one nuclei. So these are also one nuclei, just as the smooth muscle cells are. And as we can see, the appearance is a little bit different than the smooth muscle as well. So we can see that there are these striations that are going across like so. And so this is going to be a striated appearance. So striated. And then the control. And so when you think about the muscles of the heart, we don't really have to think about our heart beating. And so we know that this is also going to be involuntary control. So this is involuntary. And so the smooth muscle cells and the cardiac muscle cells have a few similarities, mainly that they have one nuclei and that they're involuntary. The main difference between those is that they are actually going to be striated with the cardiac muscle cells, but not the smooth muscle cells. All right, so let's talk about the intercalated discs. So we can see those right here connecting all of these cells together. And so with intercalated discs, we're going to have that created by a desmosome as well as a gap junction. And so desmosomes are going to be formed by cadherins and such, and those are going to be holding the cells physically together. And then we're also going to have these gap junctions. And gap junctions are basically there to connect the cells together with ion channels. And this is really important because we need all the cells of the heart to contract at the same time. And so as these gap junctions allow the ions to flow through, all of the cells are going to be able to depolarize at the same time. And that's going to allow the heart to contract at the same time as well. And so without those gap junctions, we'd be kind of a little bit in a mess. So these are really important to help that heart contract normally. Also, we can notice that the appearance of these cells is a little bit different as well. We can see they're like kind of long, and they're kind of like are connected all over the place. And so it looks kind of like a little ant farm right there. And so if you ever see cells that are kind of like in an ant farm type looking pattern, you can know that those are going to be our cardiac muscle cells. The skeletal muscle cells and the smooth muscle cells aren't going to have that appearance. And so if you see something that looks kind of like this, you can probably bet your bottom dollar that it's going to be cardiac muscle. All right, let's keep going on and talk about the skeletal muscle now. So a skeletal muscle, let's talk about these characteristics. So when you look at this, we can see that there are a lot of nuclei in each skeletal muscle cell. And so these are going to be multinucleated, so let's go ahead and write that in, so multinucleated. And the appearance we can see just as the cardiac muscle cells were striated, the skeletal muscle cells also have these striations in them. So when you're looking at these are going to be striated in appearance. Alrighty. And when you think about it, when we are doing these actions like the flexion of the biceps or the extension with the triceps brachii, those are under like our voluntary control. We have to like really think about it and like actually do it ourselves. Our body's just not going like willy-nilly, just like moving all around by itself. And so the skeletal muscles are actually under voluntary control. So this is a little bit different from the cardiac and the smooth muscle cells, which were involuntary. So voluntary control right here. And as we know, our skeletal muscles are going to insert on different bones, and basically that's really important because if we were just a skeleton, we wouldn't be able to move, and we would just be a pile of bones. But the muscles actually give us that movement that we need to walk around, and so I'm a big fan of these skeletal muscles. So just a quick recap of that. These are going to be multinucleated cells with striated appearance, and that's going to be under our voluntary control for this skeletal muscle. So let's go ahead and talk about these striations now. So a little thought question for you is why do cardiac and skeletal muscles have striations but smooth muscles don't? So just take a moment to think about that. Pause the video if you want to. But the answer is going to be right here with sarcomeres. And so sarcomeres are going to be like the functional unit of a muscle cell, all the way down to like the little basic building blocks. And basically this is going to allow for the contraction with a sliding filament theory that we're going to talk about in a few lessons. But basically the sarcomeres give that striated appearance to these muscles. And so smooth muscles will not contain any sarcomeres, but the cardiac muscle cells and the skeletal muscle cells will have these sarcomeres in them to give them that striated appearance. And so that is why cardiac and skeletal muscle cells have striations, but smooth muscle cells don't. So that's kind of a fun little thought. Alrighty, so let's go ahead and do a quick review. Hopefully you can remember all of this stuff. So with smooth muscle, let's go ahead and talk about the nuclei first. So if you remember, the smooth muscle cells are going to have one nucleus. So one nucleus in each muscle cell. And if you remember, the cardiac muscle cells also have that one nucleus in each cell. Well, with the skeletal muscle, this one does differ just a little bit. This is going to be a multinucleated cell. So this is a multi. And so these two right here, the cardiac muscle cells and the smooth muscle cells are similar in that respect, but the skeletal is a little bit different in that respect as well. Let's talk about the appearance now, and so which of these was striated and which of these weren't striated. So hopefully you can remember this. We just went over it a little bit as well with those sarcomeres. Smooth muscles are going to be non-striated, so non-striated, but cardiac and the skeletal muscle cells, those will be striated right here. So striated. Perfect, right there. And again for the skeletal muscle. All right, and so as we can see, these two are similar in that respect again. So cardiac muscle cells and skeletal muscle cells are very similar, but the smooth muscle here is that odd man out. All right, so let's go ahead and talk about the control now. So of these cells, which are voluntary and which are involuntary? So if you think about smooth muscle, those are in our organs, our blood vessels, and our airways. And so those are going to be under involuntary control because you don't have to think about using those muscles to like breathe and have blood pump through our bodies and like digest our food. And so that's going to be involuntary. So involuntary right here. And cardiac muscle cells, we also don't really have to think about our heart beating, so those are also going to be involuntary. Alrighty, and then that's going to leave us with the skeletal muscles. And if we think about it, our somatic nervous system, we have to like think about it to actually like move those muscles. And so this is going to be voluntary right there. Perfect. So as we can see here, the control for the cardiac muscle cells and the smooth muscle cells are both involuntary, so those are similar again. And that skeletal muscle is just that odd man out, and that's going to be voluntary control. All right, so as we can see, the smooth muscle cell and the cardiac muscle cells, they do have some similarities with the number of nuclei as well as the control of those cells. But they do differ in the appearance, basically that the cardiac muscle cells, just as the skeletal muscle cells, are striated, while the smooth muscle cells are not. So that's going to be a quick little review of the different types of muscles. So let's go ahead and just look at this one last time. I have a couple of review questions here I want you to think about. So if you want to just pause this, go ahead and do that, and you can think about these questions. But why do cardiac muscles and skeletal muscles have striations? And if you think about it, that's because they have those sarcomeres, and those are going to give that striated appearance. And the smooth muscle cells will not have those sarcomeres, which is why they have that smooth appearance. All right, and then what are the main characteristics of the three types of muscles? So as we know, a smooth muscle, those are going to be uninucleated. They're going to be under involuntary control, and they're going to have that non-striated appearance. With cardiac muscle cells, those are going to be uninucleated as well. They're going to be involuntary just as the smooth muscle cells are, but they will have that striated appearance. You can see those intercalated discs, and it kind of looks like that little ant farm. And then the skeletal muscles, those are going to have those striations. They're going to be multinucleated, and they're going to be under our voluntary control. And so those are like the main characteristics of those three different types of muscles. Hopefully you're able to learn from this. But if you have any questions, please feel free to reach out. We are more than willing to help you answer any questions. I want you to do well on test day, and so if you have any questions, please reach out. We would love to help you. If you don't have any questions, though, have a great day and good luck with your studies. Alright, hey y'all, we are going to be going over some skeletal muscle anatomy today. So as we can see, there are some different components that comprise skeletal muscle. So we're going to be talking about the skeletal muscle itself, which is going to be made up of these muscle fascicles. Then the muscle fascicles are going to be made up of muscle fibers, and the muscle fibers are then going to be made up of myofibrils. So let's go ahead and start talking about these guys right here. And so right here, this is kind of like a little, hopefully a little helpful picture for you. So skeletal muscle, we can see it's the largest, and then we see muscle fascicles are a little bit smaller, muscle fibers are smaller than those muscle fascicles, and then myofibrils are the smallest of all. So when you think about skeletal muscle, well, at least I do. I like think about it just one component, like one thing, but it's actually composed of smaller components, and each one just gets larger and larger as they get like built up upon. So like all these myofibrils will eventually build up into a muscle fiber. All the muscle fibers will build up into a muscle fascicle, and then all the muscle fascicles will build up into a skeletal muscle. And so you kind of think about it like a rope. So when you look at a rope or like some string, so I have some embroidery floss right here. Embroidery's one of my little hobbies that I like to do. So like when you look at this, it's just like a simple string. It's just one thing. You think it's just okay, like cool, one string. But if you pull it apart, you can actually see that there's 6 different pieces right here. And so you can actually just take that and just kind of break that apart. So it's a lot like the muscles that we have these smaller components that are like all built up into each other to lead us to the skeletal muscles that we have. So hopefully that's like a little helpful visual reminder for you that skeletal muscle is a lot more than just the muscle. It's made up of all these different components. Well, let's go ahead and talk about each one of these and what we can learn about them. All right, so as we know, skeletal muscle is going to be the largest bundle that we see. So that's what we're going to be seeing right here, what we usually see in like the gross anatomy labs. And so that's what we think about when we think of skeletal muscle. It's going to be something kind of like this. And so as we can see, it's going to be composed of a smaller component, and we can see that there's a lot of them right here in that little area. And so those are going to be composed of muscle fascicles. So I'm just going to write fascicles in here. Alrighty. And so each one of these guys in here in this skeletal muscle is going to be an individual fascicle. So we can see a fascicle right here, all that good jazz. Alrighty, so that's going to be the largest bundle. But as we can see, there is a little bit of a kind of protective barrier, not like a barrier, I guess you could say. There's kind of a sheath of sorts that surrounds the actual skeletal muscle itself. And that is going to be known as the epimysium. All right, so let's go ahead and write that in. So epimysium right here. All right, so that's going to be what surrounds the entire skeletal muscle itself. So when you think about the skeletal muscle, think about the fascicles that compose it and then the epimysium that surrounds it. So let's go on and talk about the fascicles now. So the fascicles are going to be the second bundle that kind of compose the skeletal muscle. And so we can see that right here, like I said, as well as right here. And so what we're looking at right here is an individual fascicle by itself. And as we can see, just as with skeletal muscle, it is also composed of many different components that are going to be built up. And so what are those components that are actually building those muscle fascicles up? So if you remember, these are going to be our muscle fibers. So fibers are going to build up the fascicles. All right, so just as the epimysium kind of covered the skeletal muscle and held all the different muscle fascicles together, we're going to have a similar idea for the muscle fascicles themselves. And so as we can see right here, we have the epimysium which surrounded that skeletal muscle. We can see that there's a layer around here as well that kind of does the same thing. And so this is going to be known as the paramecium. So the paramecium is going to be this portion right around the muscle fascicles that surround that. So let's go ahead and write paramecium in right here. So paramecium. Alrighty. Sweet. So those are going to be our muscle fascicles. And so just a quick little recap of this. We're going to have those muscle fascicles, which are built up of muscle fibers. It's going to be surrounded by paramecium, and then all the muscle fascicles together will go on to create the skeletal muscle. All right, so let's keep going on. All right, so let's go ahead and talk about these muscle fibers now. So this is going to be the third bundle that we want to think of. So the first bundle is going to be that skeletal muscle, then we're going to have our muscle fascicles, and then finally these muscle fibers. So that's what we're looking at right now. And so as we can see, the muscle fibers aren't the smallest unit. We're actually going to have these small guys right here. And so these guys are going to be known as myofibrils. So let's go ahead and write that in. So myofibrils. And we're going to go ahead and talk about these a little bit more on the next slide. But as we can see, just as the skeletal muscle had the epimysium, there's a little bit of a covering that surrounds the muscle fiber itself to kind of create the muscle fiber. And so this is going to be known as the sarcolemma. And so the sarcolemma is going to be the portion that surrounds the muscle fiber. So sarcolemma. All right, so just a quick recap of these muscle fibers. They're going to be that third bundle that we're seeing, and they are going to be composed of myofibrils. When all those myofibrils come together, that's what forms the muscle fiber. And that's going to be surrounded by the sarcolemma, and then a bunch of muscle fibers will then form the muscle fascicles. And as we know, the muscle fascicles, a bunch of those are going to form the actual skeletal muscle that we know. All right, so let's keep going on now and talk about these myofibrils. So the myofibrils are going to be the kind of like the smallest unit. They're actually composed of something else though, so not really the smallest, but like the smallest of what you think of with the skeletal muscle anatomy. And so let's go ahead and talk about this right here. So myofibrils are going to be composed of these contractile proteins. So there's going to be like actin, myosin. We're going to talk about that in a later video. But the contractile proteins are going to be what allows the muscles to contract. And so the fourth bundle, the myofibrils, those are going to be composed of contractile proteins. So contractile proteins, perfect. Alrighty. And so as we can see, with the muscle fiber, we had that sarcolemma. And then inside that sarcolemma, we can see that there's a bunch of empty space. And so this empty space is actually filled in kind of like by a cytoplasm type deal. And this can be known as the sarcoplasm. So instead of the cytoplasm, we're in that sarcolemma, and so that's going to be our sarcoplasm. So sarcoplasm is going to be what surrounds these myofibrils. So sarcoplasm right here. And as we can see, like with the skeletal muscle as well as like muscle fiber, there's an actual kind of sheath that surrounds them. We can see those here and right here. But with the myofibrils, there's not actually a sheath. It's just that cytoplasm-like substance known as that sarcoplasm. And so that's going to be the smallest of the units that form skeletal muscle that we're going to talk about today. And so just a quick recap of this, we can see that the myofibrils are going to be that fourth and smallest bundle. And those are going to be composed of contractile proteins that are going to work together to lead to muscle contractions. And it has no physical sheath, but it is going to have that sarcoplasm, which is going to hold the myofibrils in place. Alrighty, so let's go on to this quick review. So from largest to smallest, name the bundles in skeletal muscle. So there's going to be 4 different bundles that I want you to think of. So the first one is going to be skeletal muscle, so that might be kind of tricky because it is like the biggest one and it's right here in the sentence. So don't let that trick you up. I'm going to give you that little hint. So that's going to be skeletal muscle is the first one. So go ahead and pause this video and see if you can write in all the different types, and then we're going to go on and write about those. All right, so now that you're back, let's go ahead and write these in. So the first one, as we know, is going to be skeletal muscle. So skeletal muscle, alrighty. And then the skeletal muscle is then going to be composed of the, hopefully you remember this one, this is going to be our muscle fascicles. So muscle fascicles right here. And then the muscle fascicles are actually going to be composed of the muscle fibers. Let's go ahead and write that in. So muscle fibers. And then the muscle fibers are going to be then composed of the myofibrils. All right, so those are going to be the 4 bundles of skeletal muscle. And so we can see that it's largest to smallest right here. All right, so let's go ahead and just go on to this last review slide right here. So as we can see, the skeletal muscle is going to be composed of 4 different bundles. The skeletal muscle, muscle fascicles, muscle fibers, and myofibrils. And we have different portions of them that are going to kind of cover those areas. So we have this review question for you. So I want you to go ahead and pause this video again. Think about this review question. So can you name all the different components of skeletal muscle and what surrounds them? So with a skeletal muscle, we're going to have the epimysium, which surrounds the outer portion of that skeletal muscle. So just kind of think about that for each of the 4 different bundles. That's going to be the end of this video on skeletal muscle anatomy, but if you have any questions that you want clarified for your test day, please let us know. We want to help you in any way that we can. We're more than willing to help you answer questions that you might not have had answered or that might have come up during this video. So if you have any questions, please feel free to reach out. But if you don't have any questions, have a great rest of your day and good luck with your studies. Hey y'all, we're going to be going over the sliding filament model of muscle contraction today. And so as we can see, there's going to be 3 main things that we're going to talk about. We're going to talk about action potentials, the actual muscle contraction itself, as well as sarcomeres. So let's go ahead and get into this so we can learn exactly what all these are going to do. All right, so first off, let's go ahead and talk about this right here. So as we can see, this is going to be a motor neuron and a muscle fiber. And then we can see that there's a little bit of a space right there in between them. And so the space is actually going to be known as the neuromuscular junction. So neuromuscular junction right here. All right, and it makes sense because it's going between a motor neuron, so hence the neuro portion, and then the muscle fibers, hence the muscular portion, and it's that junction between them. It's like a synaptic cleft right there. And so that's what we're seeing right here. But because it is synaptic right there, it's going between that neuron and the fiber. The motor neurons, those are going to be known as presynaptic, so that's going to be presynaptic right there. And then the muscle fiber is going to be after that synapse, and so this is going to be a postsynaptic. And so that's going to be like the most basic kind of breakdown of the neuromuscular junction right here. It's just that space between the motor neuron and the muscle fiber, and that's going to be where all the magic happens that leads to muscle contraction. So let's go ahead and talk a little bit more about that. So what are the steps of an action potential then? So as we can see, the motor neuron is going to be the first portion because that's going to be that presynaptic portion. And so the motor neuron is going to send an action potential to the neuromuscular junction. So here it is. Let's go ahead and write that in. So it's going to send an action potential. And so we can see that when that action potential gets down to the end of the motor neuron, there's actually these little synaptic vesicles that are going to fuse with the end portion and release their contents into the neuromuscular junction. And so these synaptic vesicles, in the case of muscle contraction, is going to be acetylcholine. So that's what we're seeing right here in these synaptic vesicles. So when the action potential reaches the end of the motor neuron, acetylcholine is going to be released into the neuromuscular junction. So let's go ahead and write that in. So acetylcholine. All right. And acetylcholine is also known as ACH. So we're just going to write that down here again. So ACH is actually going to bind to a ligand that we see on the surface of the muscle fiber. And so acetylcholine is going to bind to that, and it's going to open a ligand-gated sodium channel. So there's going to be a little bit of sodium, which is going to be able to go into the muscle fiber there. And so that's going to be a ligand-gated sodium, so Na plus, because it's going to be an ion there. And that's going to create a graded potential in the muscle fiber itself. So graded potential. And so when that graded potential is entering the muscle fiber, it actually causes a depolarization or an increase in the electrical voltage of the muscle fiber to occur. So depolarization. Let's go ahead and write that in right here. All right, and that's going to open a voltage gated channel, and this is going to be for sodium again. And so it's kind of nice because both the ligand gated and the voltage gated channels are both going to be for sodium ions. And so that's just one less thing you have to remember because they're the same thing. So one is ligand gated though, and the ligand for that channel is going to be acetylcholine, and the voltage gated channel is going to be with the depolarization. So once it reaches a certain threshold, those channels will open up. And so once those voltage gated sodium channels open up, that active potential is going to be able to go along the surface of the muscle fiber, and then it's going to go down into some T tubules. So let's go ahead and talk about that process now. All right, so we've got a bigger overview right here of what we just saw on the previous slide. So let's go ahead and just talk about that real fast, just kind of review that, make sure we got all of that down. All right, so here we can see we have a motor neuron right here, and then in that motor neuron, as we know, we're going to have those synaptic vesicles full of acetylcholine. We can see the acetylcholine is right there in the neuromuscular junction, and it's going to bind right here to this little gated channel, and that's going to be a ligand gated channel, and acetylcholine is that ligand. When acetylcholine binds, sodium is then able to enter the cell, which causes a graded potential. Once that graded potential reaches a certain threshold, that's going to lead to the voltage gated sodium channels opening up, which is actually like the transfer of the active potential from the motor neuron itself into the muscle fiber itself. And then that's going to travel along here. And as we know, this is a muscle fiber, and so the outside portion of a muscle fiber we know is going to be called the sarcolemma. So let's go ahead and write that in right here. So sarcolemma. Alrighty, and as we can see, the sarcolemma is going to have these little invaginations that we see right here. And these invaginations on the sarcolemma are going to be known as T tubules. Let's go ahead and write that in as well. So T tubules. So when the action potential reaches the T tubule, it actually is going to spread through the cell very quickly, which leads to a coordinated contraction occurring. And so that's going to allow the whole muscle to contract kind of in unison, which is quite a beautiful thing. So it's just going along here. It's going to reach down into that T tubule, leading to the contraction that we all know and love, which allows us to move. So that's pretty important. All right, so let's go ahead and talk about this portion that we're seeing right here. So as you can see, this is touching the end of that T tubule. And so there's this large portion in here that has all of these ions in here. And so these ions are actually going to be calcium. And so previously we were only using sodium ions, but now we have these calcium ions. Let's go ahead and write that in. So calcium 2 plus, as we know, those ions. And then those are going to be seen in this structure known as the sarcoplasmic reticulum. And so the sarcoplasmic reticulum is basically just a specialized endoplasmic reticulum that sits in the sarcoplasm, and it's going to hold those calcium ions within that. And so this is going to be known as the sarcoplasmic reticulum right here. So sarcoplasmic reticulum. All right. And then lining the sides of the sarcoplasmic reticulum, we can actually see this portion down here, which is going to be that sarcoplasm. So let's go ahead and write that in as well. So sarcoplasm. All right, and so these are like the main portions of this. So backtracking just a little bit, we had that action potential coming down into that T tubule, and that's going to reach that sarcoplasmic reticulum. And as we can see right here, we have some gated channels. And so we don't have any ligands here, but we do have that action potential, which is going to depolarize or raise that electric potential that is in the cell. So once the action potential reaches the sarcoplasmic reticulum, that is going to allow these channels right here, which are voltage gated calcium channels, to open up. When that happens, all these calcium ions that are stored in the sarcoplasmic reticulum are able to be released right here into the sarcoplasm to then play a role on the actual contraction of the muscle. And so we've seen how the action potential travels down from the motor neuron itself through that neuromuscular junction into the muscle fiber itself, which goes over on the sarcoplasm down into that T tubule, which is going to allow these voltage gated ions channels to open up, which then releases those calcium ions down into the sarcoplasm. So that's just like a very quick overview of those action potentials. Let's go ahead and talk about what happens with the calcium now that it's kind of released and in that sarcoplasm. Alrighty, so what we're looking at right here is going to be known as the sarcomere. So as we can see, there are a couple of different parts of the sarcomere. So I want to talk about this thin filament that we see as well as the thick filament. And so these filaments are actually going to be known as myofilaments, and these are going to be composing the myofibril that we talked about in the skeletal anatomy video. And so the thin filament is going to be known as actin. So let's go ahead and write that in. So the thin filament, again, is actin. And then we are also going to have the thick filament portion of the sarcomere that's going to be known as myosin. All right, so we're going to talk a little bit more about these components later in this video. But just like a quick overview, like that sarcomere, we're going to have those thin filaments and the thick filaments. And these are going to actually connect through a cross bridge. And the myosin heads are going to pull the actin filaments towards the middle. And that's actually what leads to a muscle contraction. And so this is going to be really important to understand, to better understand how the actual muscle contraction works. And so that's going to be with the thick filament and the thin filaments, and those are going to be known as myofilaments. All right, so let's go ahead and talk about troponin now. All right, so let's go ahead and take a look right here at this image. So as we can see, we're going to have actin here, and if you remember, actin's that thin filament. We have tropomyosin, and then we're also going to have troponin itself. And so troponin, we can see that's made up of three different parts. We can see a C site, an A site, and a T site. And so each of these sites are going to bind to something different. So the T site is going to bind troponin to tropomyosin. The A site is where troponin is going to bind onto actin. And then the C site is going to be what binds to calcium. So I like to think of troponin as kind of like a staple that kind of staples down tropomyosin onto actin. And so if we look right here, we see these three sites, and we can see that it's holding this tropomyosin on. And then the tropomyosin is then holding onto the active sites, or the binding sites of actin itself. And so we can see that active site right here, or this binding site, kind of hidden by that tropomyosin. And so how does that binding site of actin actually become available? So if you look right here, we can see these calcium ions. And a little bit of a review from this last video, where do these calcium ions come from? And so these are actually going to be coming from that sarcoplasmic reticulum that we talked about. So once the action potential reaches the sarcoplasmic reticulum and those voltage-gated calcium channels open, they're released into the sarcoplasm, and then they're going to be able to bind onto troponin. So when calcium goes and it binds onto a molecule of troponin, it's going to bind onto that C site because it's going to be where the calcium binds. And when that happens, there's going to be a conformational change, and so it's going to kind of just switch itself, and that's going to release tropomyosin from actin itself. And so at that point, tropomyosin comes off of that actin, and it's no longer in contact with troponin either. And the actin is then free, and we can see right here that there's this active site. And so that active site is finally open. So let's go ahead and write that in. So active or binding site. All right. And so we can see right here that that actin is then free. All right, and so we had talked about that thick filament as well, and that's going to be myosin. And the myosin heads are actually going to want to bind to actin itself. But until these binding sites are actually made free, it can't do that. And so a little fun fact here, the resting state of a muscle is actually going to be contracted. But when we're in that relaxed state, those active potentials aren't going through because there's no neurological signal to actually contract that muscle. And so the resting state is contracted, but it can't actually be contracted because these guys right here, these binding sites on actin, are actually unavailable to the myosin heads to actually contract the muscle. And so always remember that the resting state of muscle is contracted, but it can't do that because we don't have those active potentials being sent. And so once that active potential is sent, the active sites are then available, and then the myosin heads can actually pull on the actin to make that muscle contraction happen. And so what happens when calcium binds to troponin? Basically, the troponin is then going to have that conformational change. Tropomyosin is going to be released off that actin as well, and then those actin binding sites are then free for the myosin heads to actually make that contraction happen. So that's a little bit of an overview of what happens to calcium when it binds to troponin. All right, so let's go ahead and talk about what actually happens here. So there's four different steps we want to talk about. But first off, let's take a look at these real fast. So we can see right here that these are pointed to the actin filaments. And so let's go ahead and write actin in one last time right here. So actin. Then right here we can see myosin, and we can see the myosin heads right there with this ADP and this phosphate. So let's go ahead and write in myosin right there. So myosin. All right, so these are going to be our thick filaments and then our thin filaments. And these will be interacting to actually make that muscle contraction occur. So right here, let's go ahead and talk about step one. And so a fun little fact about these guys right here, these myosin heads, these are actually ATPases. And so that's going to hydrolyze ATP into ADP and an inorganic phosphate. And so that's what we see right here. And so this hydrolyzation is what is occurring there. And when ATP hydrolyzes, that's going to be a really big release of energy. And so this is going to be very energetically favorable. And so this is what happens in that first step. And so basically myosin splits the ATP, and that leads to a large energy increase, which helps reorient the myosin head to properly attach onto actin. And so right here we can see that ATP going to ADP and that inorganic phosphate, and then the myosin head is also reorienting itself. And then we're going to have step 2 right here. And as we can see, the myosin head is not actually attached to the actin right here, but in this one it is. And so this is actually when that cross bridge happens. So this is when the myosin head is going to bind onto the actin. And so this is when the thick filament and the thin filament are interacting with each other. And we call this the cross bridge formation. So this is when that cross bridge happens on that second step. And so basically the myosin head is then attaching onto the actin. And so those two filaments are then interacting. Here between step 2 and step 3, we can see that the ADP and the phosphate are kind of kicked off. And so this is actually going to be when the actin is being pulled by the myosin heads. And so when this happens, it's actually known as the power stroke. And so the power stroke is what happens right here. And so we can see that the actin and the myosin are in a different position here than they are here. And that's because that myosin head has pulled that actin closer towards that middle or the M line of the sarcomere to make that muscle contraction occur. And so right here, there's no ADP, there's no inorganic phosphate because those have been kicked off right here with that power stroke. And so that muscle is stuck in that contracted state. All right, and so then the final step for this all 3 set, for that myosin head to be released from the actin, we need another molecule of ATP. And so when that ATP is going to bind to the myosin head again, that's actually going to cause the myosin head to release the actin that it's holding onto. And that's going to be the end of the muscle contraction right here. So you can see once again right at this step that the myosin head is no longer attached to actin, that cross bridge is broken, and then the molecule of ATP is still attached to myosin. So we know that this cycle goes on and on and on until that nerve impulse stops sending that signal. And so this cycle is just going to keep going on and on. And so when that nerve impulse stops, that is when the sarcoplasmic reticulum, those voltage-gated calcium channels, are going to close, but that calcium is still in the sarcoplasm itself. And so the calcium actually has to be actively transported back into the sarcoplasmic reticulum. And so all that calcium that was once bound to troponin is going to be then detached from the troponin, and then it's going to go back up into that sarcoplasmic reticulum for the next nerve impulse to fire. And so if you go back to this picture right here, we can think about this. And so if that calcium is no longer on that troponin, that's going to go back to its original conformation. That's going to then bind tropomyosin again, and then the tropomyosin is going to kind of cover that binding site and hide it so that the myosin heads can no longer attach to actin. And so when that calcium is gone, it's going to return back to this original state, and no muscle contraction will be able to occur even in that relaxed state, because the binding sites on actin are then covered again. So that's kind of why our muscles do stop contracting after those nerve impulses have stopped. They don't just keep going on and on. That's why they stop right there. So let's go ahead and talk about rigor mortis right here. And this is an applicable concept with this ATP. And so rigor mortis is when we get really stiff after we die. So this happens in animals and humans and everybody. And so I want you to just pause this video and just think about why that might happen. Think of a reason why. Hint, it has to do with ATP. And then when you think you might know why rigor mortis occurs, go ahead and start this video again. But basically it's going to occur because we don't have any ATP left after we're dead, because our bodies are no longer producing ATP. And so the myosin head and the actin filament, they can no longer detach. So the cross bridge is still formed and it's in that contracted position, which is why we get really stiff, and that's what rigor mortis is. And so basically the gist of it is that we don't have any ATP to actually make that cross bridge detach from each other. So the actin and the myosin, those filaments are just stuck together in that contracted position. And so that's going to be a little bit of an explanation for rigor mortis. And it kind of like ties into all of this with that understanding that ATP is going to make that cross bridge detach. So just a little quick overview from everything. We have that action potential that goes from the neuron into the muscle fibers. That depolarization and graded potential occurs, which leads to the depolarization into the sarcoplasmic reticulum, which will then release those calcium ions, which then bind to troponin and do that conformational change to release tropomyosin from actin itself. And then the actin is then able to bind with the myosin heads, and this whole cycle occurs right here. Alrighty, so as we know, we have the sarcomere right here, and we've talked a little bit about this before. We've talked about those thick filaments and the thin filaments, but we haven't talked about these portions down here, the zones and the bands, and then we haven't talked about the lines either. So let's go ahead and just label this really quickly, and then we're going to go through and talk about some helpful hints to help you remember which one is which, and hopefully it'll help you atom test it. So let's go ahead and just go through and label this. So as you know, this whole portion right here is one sarcomere. And so this portion right here, right in the middle, is going to be known as the M line. So middle for M, which is always kind of nice because it's just nice and convenient for us. So that's going to be the M line right there. And then at the ends right here, I really like to use alphabetical cues to help me remember things. And so since this is at the end of the alphabet, we have the letter Z, and so this is going to be our Z line. So let's go ahead and write that in as well. So M line and Z line is the one on the end. All right, then we're going to have a band, we're going to have some zones. So when we have this, we're going to have an A band, we're going to have an I band, and they're going to have an H zone. So the I bands are going to be right here where there's only going to be actin filaments. So that's going to be the I band. So I band is right here. Then we're going to have our H zone, and that's going to be where there's only going to be myosin filaments right there. So let's go ahead and write this one in as well. So that's going to be our H zone. And then this portion down here, we can see that that's going to line up perfectly with those myosin filaments. So that's going to be whether there is only myosin or there's myosin and actin filaments as well. So it's going to be the whole length of the myosin filament is going to be known as the A band. So let's go ahead and write that in as well. So A band right here. Alright, so that's going to be the different portions of the sarcomere right here. So we have that M line like we know, the Z line. They're going to have our H zone right here, which we can see right here, right here, and right here. We're going to have our I bands, which are going to be these portions right there. And we're going to have our A band, which is going to be right here. Alright, so let's go ahead and talk about some helpful hints to help you guys remember these, because we want you to be able to do well on test day, and these will likely come up. So make sure you understand this really well. Alright, so I like to use the placement of the lines to kind of help me remember for the names of the lines. So like I was talking about before, we have this one right here in the middle. So that, of course, is going to be our M line. And then these ones are at the ends. And so at the ends of the alphabet, we have the letter Z. So we have our M line and our Z line right there. So I'm just going to go ahead and write these in real fast. We're just going to put M, and then we're going to put Z right here next to the end ones. And then we're going to use the thickness of the letters to know the bands and the zones. So we're going to have two bands and one zone. So let's go ahead and start with this one right here. This is where there's only going to be thin filaments. And so the letter I is a fairly thin letter. And so I like to think that we can use that letter, the letter I right there, to help us remember that's going to be the thin filaments only. And then this one right here, this is where there's only going to be thick filaments. And H is a little bit of a thicker letter. I is a little bit skinnier, H is a little bit thicker. And so we know that this one right here, where there's only thick filament, that is going to be known as the H zone. So let's go ahead and write that in as well. So H right there. And if you remember this one right here, that's going to be our A band. And our A band is going to be the length of the entire thick filament, whether that is the thick filament only, or whether it's also with the thin filament as well. So that's going to be our A band. So let's go ahead and write that in right here. So A. Right, and like I said, I do like these alphabet cues. And so right here and right here, we have those vowels, so A and I. And these are both going to be bands, while this one right here, the consonant H, that is going to be a zone. And so if you ever can't remember which one's a band, which one's a zone, just remember that the vowels are both going to be those bands, and then the consonant is going to be the zone right there. And so hopefully that can be helpful on test day if you kind of get into a bind right there and you're just thinking about it. Hopefully that can help you remember those. But like I was saying, we're going to have our M line, our Z line, our H zone, and our I band, and our A band right there. And those are going to be the different components of the sarcomere. Something that's kind of like interesting to note is that when the actin filaments are sliding towards this M line, like so, as we can see with these arrows, both the H zone and the I band are going to get smaller because the actin filaments are going to be sliding this way, which will make this smaller. But at the same time, those myosin filaments are getting closer to this Z line, making this portion smaller. And so those are both going to shrink during a muscle contraction. But right here with the A band, this is just the length of the entire myosin filament, whether there's actin there or not as well. And so the A band, that is never going to change in size. It will always be constant throughout the entire muscle contraction. So that's going to be how the sliding filament theory explains muscle contraction. So let's go ahead and just do a quick little review right here. So we've talked about all of these different portions during this video and the previous video. And I have a couple of review questions that I want you to think about. So right here, we're going to have these two questions. So we have the question, can you describe how an active potential is translated into a muscle contraction? So basically that's like the whole outline of what we just talked about in these two videos. So it's a pretty long process, but if you can explain that to like a friend, a roommate, a family member, whoever you want to explain it to, that will help you. First, that'll help you get it down into words, and it'll also help you remember it better because you've been able to teach it to someone. So that's what I would recommend doing for this first review question. And for the second, what are the different elements of the sarcomere? And so for this one, I would just recommend drawing it out. So you can just do a simple drawing just like this. And then we have these guys right here going like so. That's a very crude drawing of it, of course. I'm not the best artist that quickly. But if you can do that and then label these guys right here with the M line, the Z line, the H zone, the I band, and the A band, I would say that you're in really good shape. All right, and that's going to be it for this video. But if you have any questions that we didn't answer, or you have any questions about some of the concepts that you didn't quite understand, please feel free to reach out. We want you to do as well as possible on test day. We're always here to help you. But if you don't have any questions, have a great rest of your day and good luck with your studies. Hey y'all, it's Lance, and today we are going to be talking about motor units. So motor units, we're going to have small motor units and large motor units. So let's go ahead and just jump into this so we can learn more about what motor units actually are. Alright, so first off, let's go ahead and start with a thought question. So based on this image, what is a motor unit? So as we can see from this image over here, we can see these neurons going to different muscle fibers. So go ahead and just pause this video, think about what a motor unit actually is. You might know already, you might not know, but based off this image, try to come up with a functional definition of what a motor unit is. All right, so now that you've had some time to think about that, let's go ahead and talk about what it actually is. So as you can see, we have a neuron right here, and it's going to a few muscle fibers right here. And then we have another neuron right here, which is also going to some muscle fibers. And so in this image, there are actually two motor units. And so there's one right here, and then there's going to be one with these red fibers that we see. All right, so based off of that, we can see that there's going to be one neuron. So this is going to be one neuron. And then it's going to be all of the muscle fibers that it is going to innervate. So all muscle fibers. Alrighty. So this right here is going to be that functional definition of what a motor unit is. So with this example over here, we can see these blue fibers, so right here and right here. And as we can see, this neuron is innervating those fibers, and so that entire thing would be one motor unit. All right, so let's go ahead and talk about these motor units now because we have large motor units, as we can see, and small motor units. So let's go ahead and start with these small motor units. And so small motor units are going to be one motor neuron, and as we can see, it's just going to just a few little fibers right there. And so we know that this is going to be just a few muscle fibers for the small motor unit. So few muscle fibers. All right, so this is going to be that definition for a small motor unit. And so what kind of movement do you think it's going to produce? Do you think it's going to be very precise? Do you think it's more powerful? So go ahead and just think about that for just a second. Pause the video if you need to. But these are actually going to be more precise movements because it's just a few muscle fibers. It's not a lot. And so these are going to be very precise movements. Let's go ahead and write that in. So precise. Alrighty. So now that we know the characteristics of these small motor units, let's go ahead and think about what muscle has small motor units. So go ahead and pause the video and think about that based off of your anatomy knowledge. So there's going to be some muscles in the body that have small motor units that really work with those. So just go ahead and think about that for a second, and then we'll come back to this. But basically, the one that comes to mind first, for me at least, are going to be like the muscles of the eye. So an example of that would be like the superior rectus. And so the superior rectus is going to have very precise movements to help move our eyes to the right, to the left, up and down, whatever movement that we need to have with our eyes. Those are going to be very precise movements, and those are going to be innervated by these small motor units. All right, so let's go ahead and start talking about these large motor units now. So we know that the small motor units are going to be one motor neuron and just a few muscle fibers. But as you can see from this image, we can see that there's one neuron that goes to quite a few more muscle fibers than these small motor units do. So this is going to be one motor neuron and many muscle fibers, so many right here. And if you think about it, when we have more muscle fibers contracting at once, is that going to be more precise or more powerful movement? That's right, it's going to be more powerful because you have more fibers contracting at once. And so these are going to be more powerful movements. So let's go ahead and write that in. So powerful. Alrighty. And so now that we know these characteristics, go ahead and pause the video and think about what muscle has a large motor unit that you can think of quite readily. All right, so now that you've had some time to think about that, the muscle that comes to mind first for me is going to be the rectus femoris or those other quadriceps femoris muscles. Just because they're really large, they're going to provide very powerful movements for us as we're walking, as we're running, doing everyday things. And so that's going to be a muscle that I think of that has large motor units. So when I think about small motor units and large motor units, I kind of have them like very separate in my mind for some reason. But when you think about it, they're actually going to be used together. So I really like to ski, and so when I'm skiing, I'm looking around with my eyes. So that's going to be those small motor units that are providing those very precise movement of my eye. So I can see where I'm going so I don't crash into a tree or into another skier or a snowboarder. So those are going to be very helpful in that. But I'm also going to have these muscles that are innervated by large motor units, like my rectus femoris, my biceps, brachii. All these muscles are going to be innervated by large motor units, and those will help me move my poles correctly, move my skis correctly so that I can actually get down the mountain without falling. And so these small motor units and these large motor units are all working together to help me get down the mountain in one piece. And they're also going to be helping us get around town when we're driving, when we're doing everyday things. And so these small motor units and these large motor units really work together to help us accomplish our everyday tasks. All right, so that's going to be it for this video. So we've just talked about all these things over here. So the motor units, we have that functional definition being one motor neuron and all of the muscle fibers that it innervates. And then you talked about these small motor units and these large motor units as well. So I have a review question right here for us. And the review question is, what are the key differences between small motor units and large motor units? So I want you to think about how many fibers are in each one, what kind of movements they're going to produce. So go ahead and think about those differences between the small motor units and the large motor units. And hopefully this video will have helped you get those down because these will be important for test day. But if you had any questions that came up during this video, or if you have any questions that weren't answered during the video, go ahead and reach out to us. We are more than willing to help you answer those questions so that you can do as well as possible on test day. But if you don't have any questions, have a great rest of your day and good luck with your studies. Hey, it's Lance, and we're going to be talking about twitch contractions today. So if you want to go ahead and pause this video, you can look at the outline for what we'll be talking about specifically, but we're just going to go ahead and just jump right into it. Alright, so what is a twitch contraction? So if you want to go ahead and just pause this video and think about what blank is probably going to be filled in here, whether there's going to be a single action potential, whether it's going to be multiple action potentials, and see if you can fill this in, and we're going to go ahead and talk about this in just a second. So now you've had some time to think about that, let's go ahead and fill this in. So a twitch contraction is actually going to be just one single action potential, so a single action potential right here, perfect. Alrighty, and so this is going to be a simulation of an entire motor unit. So if you remember, a motor unit is going to be one motor neuron and all the muscle fibers that it's going to innervate. So a single action potential that we see right here, that's going to go ahead and travel through that motor neuron, and then that's going to stimulate the entire motor unit to result in that twitch contraction. So a very brief contraction of that muscle right there. And a really important concept with this is going to be the all or nothing principle. Alright, so with the all or nothing principle, we can see that there's depolarization that occurs, and when that depolarization reaches threshold, so anything above threshold or at threshold, that will result in a twitch contraction. But if it is below that threshold, there will not be a twitch contraction because the action potential will not send. And so basically, if that depolarization happens enough, then it's going to send, but if it's below that threshold, that's not going to result in a twitch contraction. So once again, just to reiterate this, because it's a really important principle and it will likely show up on test day, if it is above that threshold, that will result in a muscle contraction because it's depolarized enough. But if it's not depolarized enough and it's below that threshold, there will be no twitch contraction. And so you might be wondering, if it's super high above that threshold, is that then going to result in like a stronger contraction, or is it going to be the same? And it's actually going to be the same because it just has to reach that certain threshold in order for the action potential to send down into that muscle fiber to innervate that motor unit. And no matter how high that depolarization is, or just if it's just barely above that threshold, they're going to result in the same force of contraction. So no matter where it's at above this threshold, that's going to result in the same strength of twitch contraction. And so that concept holds true with one single muscle fiber. So if it's above this threshold, it's going to have the same size and duration of a twitch, but that might not translate into exactly the same for another muscle fiber near the other muscle fiber or in a different muscle itself. So basically, each muscle fiber separately is going to have the same size and duration of twitch contraction, but a neighboring muscle fiber might not. It just depends on that muscle fiber itself. But remember, each muscle fiber is always going to be the exact same every single time as long as it reaches this threshold. Alrighty, so let's go ahead and talk about the actual twitch contraction itself now. So as we can see, there are going to be three different phases. So we can see one phase right here, one phase right here, and one phase right here. And so these three phases are going to have different names. So let's go ahead and write all of these in. So this first one that we're seeing right here, this yellow, this can be known as the latent phase. So go ahead and write this in. So latent phase right here. And then the second phase that we can see right there in red, that's going to be known as the contraction. And then the third that we see right here in blue, this is going to be known as relaxation. Perfect. Alright, so these are going to be the three phases of a twitch contraction, and we can see those right here. So let's go ahead and talk about each one of these individually so we have a better understanding of each one. Alrighty, so let's go ahead and start with the latent phase because that's the first one. We can see that right here on this little graph. And so we can see that this is the very first portion right there. It's not super long, and we can also see that there's not a lot of tension associated with this phase. So it starts out at zero tension because there's nothing happening. And that's going to lead into this portion, which we'll talk about in just a second. So let's go ahead and talk about what is actually happening in this. Alright, so if you remember from the sliding filament theory, we had this little drawing in here. And so as we know, the action potential is going to reach the sarcolemma right here. The acetylcholine is going to bind onto these ligand-gated sodium channels, and that's going to lead to a depolarization that occurs, which will eventually reach this portion down here. If you remember, that's going to be the sarcoplasmic reticulum. And in here, we're going to have all of these calcium ions. And so in the latent phase, that action potential is going to send, and remember with the all or nothing principle, it has to reach that certain depolarization for the action potential to send. But if that potential is reached, all these calcium ions will then be released, and those will go down into the sarcoplasm, which we see right down here. And so that's what's happening in the latent phase, is basically the release of these calcium ions from the sarcoplasmic reticulum down into the sarcoplasm. So that's going to be what is happening in the latent phase. So now let's go ahead and talk about contraction. So contraction, as we can see right here, this is the actual contraction of the twitch itself. And so that's going to be this red portion. So as we can see, it's a lot longer than the latent phase itself, but it's not quite as long as the relaxation phase. But with contraction, that's going to be when the sliding filament theory is actually occurring. So when those myosin heads are interacting with actin. And so let's go ahead and talk about that a little bit more in depth. And so we've talked about this image before with the sliding filament theory, and so let's go ahead and just kind of touch on this again, give you a little bit of a refresh if you've forgotten it, or hopefully you can teach it to yourself again real fast if you remember it as a little learning exercise. So right here we can see that calcium that was released into the sarcoplasm in the latent phase. And so as we can see, we're going to have calcium ions that's kind of floating around in that sarcoplasm. And right here we have that C site on troponin itself. And so when calcium comes down and binds onto that, we can see that the tropomyosin and the actin, those filaments are going to be released from the troponin, which will make those binding sites, which we see right here on actin, available. And so when those binding sites are then available, the cross bridges are going to form between the myosin filaments and the actin filaments, and that's when the sliding filament theory occurs. And so the myosin is going to grab onto the actin, pull it closer to the M line, and that H zone that we see in the middle, that's going to start to shrink because those myofilaments are being pulled closer together. And so that's just a little quick refresher of what happens during that contraction phase when the sarcomeres are beginning to pull together. When those cross bridges form. All right, so let's go ahead and talk about this phase right here, this relaxation phase. This is the last phase of a twitch contraction. And so this is when the calcium ions are being transported back into the sarcoplasmic reticulum from the sarcoplasm. So let's go ahead and take a look at this. So as we can see right here, we have all these calcium ions that are in the sarcoplasm, and a lot of these are going to be bound to the troponin itself. But there's going to become a time when these calcium ions are going to be pumped back into the sarcoplasmic reticulum, and there's going to be an active transport of these calcium ions back into there. And so when the active transport happens, these calcium ions are then going to move up back into that sarcoplasmic reticulum. They will be taken off from those C sites of troponin, which will then bind tropomyosin again, and actin, and that's going to hinder the cross bridges from forming, and so no more contraction is going to happen, and that's going to result in this relaxation that we know. And so those fibers are going to stop contracting and they're going to relax because this calcium that we see is no longer in the sarcoplasm, but it's going to be in the sarcoplasmic reticulum instead. And so those are going to be the three phases of a twitch contraction: that latent phase, the contraction, and the relaxation. Alright, so let's go ahead and just do a quick little review right here. So we have the twitch contractions. And as we remember, a twitch contraction is when there's going to be a single action potential that is going to innervate an entire motor unit. The all or nothing principle is going to have to reach that threshold for the action potential to actually cause a motor unit to contract. And then we're going to have these three phases of contraction, which kind of leads into the review question that I have for this video. So what happens in the three different phases of a muscle contraction? So I want you to think about the specifics of the physiology behind each of these phases. So what happens in the latent phase itself, what happens in the contraction phase, and then what happens in the relaxation phase. If you can understand the concepts behind all of these and what happens on a very molecular level, you'll be in a good spot. If you have any questions about that, or you didn't fully understand something, or you had a question come up during this video that you didn't have before, please feel free to reach out to us. We want to help you do well on test day, and we want you to succeed. But if you have any questions, have a great rest of your day, and good luck with your studies. Hey guys, it's Lance, and today we are going to be talking about types of muscle fibers. So let's go ahead and talk about those. Those three are going to be slow oxidative fibers, fast oxidative glycolytic fibers, and fast glycolytic fibers. So let's go ahead and dive into each one of these so we can better understand the main characteristics of each one of these types. Alright, so first off, let's go ahead and just name those types of fibers again so we just went over them. Hopefully you can remember them, but if not, that's alright. So the first one that we're going to talk about is going to be number one. This is going to be our slow oxidative fibers. So slow oxidative. And these are also known as type 1. So type 1 right there. Alrighty, and then our second one is going to be our fast oxidative glycolytic fibers. So fast oxidative glycolytic. And these are going to be type 2A. So type 2A. Getting close to that wording. Alrighty, then we're going to have our third type of fiber. And this is going to be fast as well. And this one's just going to be glycolytic, however. So glycolytic right here. And this is going to be type 2 as well. So both of the fast are going to be type 2s, but this is going to be type 2B instead of A. Alright, so these are the three types of fibers with those secondary names, type 1, type 2A, and type 2B. And then let's go ahead and think about this real fast. So why do these fibers contract at different speeds? So obviously they are at different speeds because we have slow, we have fast, etc. And so the hydrolysis of ATP is going to occur at different speeds. And so we're going to have different speeds of that hydrolysis. And so that's going to result in the different contraction speeds. So that's actually kind of cool. It kind of like ties in the sliding filament theory with those myosin heads and the cross bridge formation. So it's kind of a cool little fact about these different types of fibers and why they contract at different speeds. All right, let's go ahead and answer these questions now. So let's go ahead and do this little exercise right here. So go ahead and match the type of fiber to speed of contraction. So if you want to just pause the video, try to figure this out on your own at first, you can. We're going to go ahead and just answer this real fast. So first off, let's talk about the slow oxidative fibers. And so since that has slow in the name, this is obviously going to be the slowest. And I'll talk about why this is in just a little bit. And then we're going to have our fast oxidative glycolytic fibers. And we also have our fast glycolytic fibers in general as well. But this one actually, the fast oxidative glycolytic fibers, these are going to be at an intermediate speed. So that's what we're going to see right here. And that's going to leave the fast glycolytic fibers to be the fastest contractions. So as you remember from that last slide, these contractions occur at different speeds because the ATPases hydrolyze at different speeds. And so the ATPases on the myosin heads for the fast glycolytic fibers are going to undergo that hydrolysis. So that's why this one's going to be the fastest and this one's going to be the slowest because of those ATPases. Alright, so let's go ahead and talk about the specifics of these guys right here. So do you remember which type of fiber this is? So is it type 1? Is it type 2? So go ahead and think about that real fast. But as a reminder, this is going to be a type 1 fiber. So let's go and write that in. So type 1. Alright, so now let's go ahead and talk about these characteristics. Alright, so for the contractions, these are going to be with the strength of the contractions. As it is a very slow fiber, this is going to be a very weak contraction. So let's go ahead and write that in. So this is going to be weak. And this is also going to be due because it has a very small diameter, and so they're not going to have a lot of force behind those contractions anyways. And so these slow oxidative fibers are going to be very small in diameter, leading us to these weak contractions. Alrighty, now the color. And so you might think, oh, like all muscle fibers are going to be red. There's actually going to be white and red fibers. And so are these slow oxidative fibers going to be red or are they going to be white? And so in this case, these guys are going to be red. So let's go ahead and write that in. All right, so why are these guys red? That's going to be because they have a really rich network of capillaries. They're going to have a lot of mitochondria. They're going to have a lot of myoglobin in them, which accounts for this red color that we see that is characteristics of these slow oxidative fibers. So when you think about it, since it has all these blood vessels in them, the oxygen levels are going to be fairly high. So we're going to have high oxygen levels, and those are going to be brought in through those capillary systems that are in these oxidative fibers. And as we know, aerobic respiration happens because oxygen is there. And so ATP is going to be produced through aerobic oxidation, which accounts for the high levels of mitochondria that are found in these fibers as well. Alrighty, so now let's talk about the twitch duration and the efficiency of these fibers. And so I kind of like to think of it as a little kind of a scale. And so these have very weak contractions, but since it is weak, they're going to be able to contract for longer periods of time. So the twitch of these are going to be the longest twitch right here. So it's going to be the longest, and it's also going to have a very efficient twitch. It's going to resist a lot of fatigue. And so this is going to be the most efficient and resistant to fatigue. Alrighty, so let's go ahead and just recap this real fast. So for slow oxidative fibers, they're going to have weak contractions, and as you remember, that is because they have that very small diameter. They're going to appear red in color because they have those blood capillary systems. They're going to have a lot of myoglobin. They're also going to have a lot of mitochondria, which leads to this red color. Oxygen levels are going to be high because it undergoes aerobic respiration. It's going to have a long twitch duration, and it's also going to resist fatigue fairly well. And as a reminder, these are the type 1 fibers or the slow oxidative fibers. So let's go ahead and talk about the fast oxidative glycolytic fibers now. Alright, so hopefully you can remember which type of fiber this was from the beginning of this video. But if you don't remember, that's alright. So slow oxidative fibers were going to be those type 1 fibers. And this is going to be a type 2 fiber. And is this one going to be A or B? That's going to be A, right? So this is going to be type 2A fibers, or those fast oxidative glycolytic fibers. So now let's go ahead and talk about these characteristics. They are going to be slightly different from the slow oxidative fibers, but there will be some similarities as well. So the slow oxidative fibers, as you remember, those had those weaker contractions because they had that small diameter. But the fast oxidative glycolytic fibers, they are more intermediate in diameter. They're a little bit larger. And so these are going to have strong contractions. So they're not going to be weak. They're going to be very strong. So let's go ahead and write that in. So strong contractions. Alrighty, and as we can see from the name, there's going to be some aerobic respiration that occurs to produce that ATP. And so as we know, that's going to require a lot of oxygen. And so these are also going to have a lot of blood capillaries. They're going to have that myoglobin to hold on to that oxygen. They're also going to have a lot of those mitochondria. Those levels won't be as high as they were in the slow oxidative fibers, but they will still be present in these type 2A fibers. So the color on these are also going to appear red because they have those blood vessels, that myoglobin, and the mitochondria. So these are going to be red in appearance as well. All right, and as we remember, we're going to have those oxygen levels because we do have that oxidative aspect to these fibers. And so these are going to be at about a medium oxygen level, so medium right here. All right, so not quite as much as those slow oxidative, but still a fair amount. All right, so as you can see, we're also going to have this glycolytic portion to this fiber. While it undergoes aerobic respiration, it's also going to undergo anaerobic respiration. And so for anaerobic respiration, we're going to have stores of glycogen within the muscle itself. So the fast oxidative glycolytic fibers are going to store glycogen. So let's go ahead and write that in. So right here, glycogen. Alrighty, and like I was saying with these contractions, so we had like the really weak contraction for the slow oxidative, but they had a longer duration. And so this one's a little bit higher up on that graph, so it's going to have an intermediate duration because it has that strong contraction, but it's not quite the strongest contraction of them all. So it's going to have that intermediate length. So let's go ahead and write that in. So it's going to have an intermediate. Alrighty, now let's go ahead and talk about that efficiency. And so while these fibers are fairly intermediate of a duration. They are still going to be fairly efficient, but they will be susceptible to fatigue. So we can't use them for quite as long of a period as we can those slow oxidative fibers. But they're still fairly efficient and resistant to fatigue, but they will be susceptible to fatigue at a certain time. So susceptible to fatigue, but those still will be efficient, like I was saying. Alright, so these are going to be our fast oxidative glycolytic fiber characteristics. So like I was saying, we have those strong contractions. They're going to appear red in color because they do rely on that aerobic respiration. And so they're going to have those blood capillaries, they're going to have the mitochondria, they're going to have myoglobin. We're going to have medium amounts of oxygen because we're also going to have glycogen stores for the anaerobic respiration of these fibers. The twitch duration is going to be an intermediate twitch, so it's going to be shorter than the slow oxidative fibers, which is going to be longer than the fast glycolytic fibers, and then it's going to be susceptible to fatigue, but it is still fairly efficient. So these are going to be those type 2 fibers or those fast oxidative glycolytic fibers. Alright, so let's go ahead and talk about the last type of fiber now, the fast glycolytic fibers. So let's go ahead and start filling these guys in. So the fast ones, as you remember, are going to be type 2 fibers. So that's what we're seeing over here. So type 2, and if you remember right, we're going to have the type 2A are going to be this fast oxidative glycolytic, and so the fast glycolytic fibers are going to be type 2B. Alrighty, so let's go ahead and start filling these characteristics in now. Alrighty, so the fast glycolytic fibers, these are going to be really big in diameter, and so these are going to have the most powerful contractions of the three types of fibers. So these are going to be very powerful contractions. Alright. And these colors, as we know, the fast oxidative glycolytic and the slow oxidative fibers, those both appeared red in color. These are going to appear white because we're not going to have those blood capillaries. We're not going to have the high amount of myoglobin to hold on to the oxygen. We're not going to have the high levels of mitochondria. They will still be present because we know all fibers do need a little bit of a blood supply, but they're not going to have the high levels, and so these are actually going to appear white rather than red. So these fibers will be white. Since we don't really have that capillary network that we had in the other types of fibers, the oxygen levels are going to be quite low in these types of fibers right here. So these are going to be low levels of oxygen, but it's also going to store glycogen because this is going to be undergoing anaerobic respiration with the glycogen. So this is going to have high levels of glycogen. So high levels of glycogen. Alrighty. And twitch duration. So as you remember with that little like kind of inverse chart, when you have really weak contractions, we're going to have long durations. And so since this has a very powerful contraction, this is going to be a very short duration. And so this is going to be the shortest of them all, right there. And so another little pattern that we can see, the shorter the twitch duration, the less efficient it is. So fast glycolytic fibers really aren't super efficient, and they fatigue very quickly. So let's go ahead and write that in. So fatigue rapidly. All right, so let's go ahead and talk about these characteristics to make a nice little picture for us all. So we have these fast glycolytic fibers or these type 2B fibers. They're going to have very powerful contractions, and as you remember, this is going to be the fastest type of contraction as well. They're going to appear white in color because they don't have those vast capillary networks. They're not going to have a lot of myoglobin or a lot of mitochondria, so they're going to appear white. The oxygen levels are going to be low because we're not going to undergo aerobic respiration here. We're going to undergo anaerobic respiration to create ATP. And so that's where we're going to have a lot of glycogen storage in these fibers. They are fast twitch fibers, but they are very short in duration, and they aren't very efficient because they're going to fatigue rapidly. And so even though they have very powerful contractions, they are going to fatigue rapidly. And so there's kind of like that trade-off there. But just as a review, those are those fast glycolytic fibers or those type 2B fibers. All right, so let's go ahead and fill out this little chart right here as a little bit of a review. Hopefully you can remember all of these from the video itself. But if not, go ahead and rewind this video, kind of think back about what you don't know on here so you can kind of get this down. This will be really important for test day to be able to differentiate between the different types of fibers and the different characteristics. So hopefully at the end of this video you can fill this out. If not, I would recommend rewatching it just so you can get those questions answered and get this information down for test day. All right, so first off, let's go ahead and go back to the very beginning and talk about the slow oxidative fibers or the type 1 fibers. So let's go ahead and just write these in real fast. So the color of these slow oxidative type 1 fibers, is going to be red in color, so red right here. Respiration type, this is going to have high levels of oxygen, so this is going to be aerobic right here. Alrighty. And the contraction strength of these is going to be fairly weak, but as we know, that's going to be a more efficient type of contraction. So it's going to be a weak contraction like we know, but in return, that's also going to be resistant to fatigue, so it's kind of a trade-off right there. So resistant to fatigue right here. All right, so now let's go ahead and do this same thing for the fast oxidative glycolytic, or the type 2A fibers right here. So as you remember, this one's also going to appear red. The levels of the blood capillaries, the mitochondria, the myoglobin will not be as high as they are in the slow oxidative fibers, but they will still have a fair amount in these other fibers. All right, so the respiration type, these are going to be aerobic respirations as well as anaerobic respirations. So aerobic and anaerobic. Perfect. Alrighty, and then the contraction strength of these is going to be a fairly strong contraction. So it's not like the strongest of all of them, but it is fairly strong. So let's write in strong right here. And as we know, these are going to be fairly efficient as well, but they will be susceptible to fatigue. So let's just write that in. So susceptible to fatigue. Alrighty. So now we're going to have this last one, the fast glycolytic type 2B fibers. And so this one differs from the rest in that it appears to be white instead of red. So it's going to be a white appearance. This one's going to be solely anaerobic. It's not going to have enough oxygen levels to produce ATP from aerobic respiration. So it's going to rely completely upon glycogen for the anaerobic. Alrighty. And then this contraction strength right here is going to be a very powerful, very strong contraction. So the strongest of all three types of fibers. So strongest right here. But it does come with that trade-off, which is going to be not very efficient at all. It's going to fatigue very rapidly. So rapid fatigue right there. Alrighty, so this is going to be a very quick overview of all of those fibers right there. So hopefully you can fill this chart out on your own. If not, go ahead and rewatch this video, kind of really get this down. This will be really important on test day. And so make sure you're able to understand all of these different characteristics and why they are the way they are. Alrighty, so let's go ahead and just do this quick little review. I have some review questions for you right here. But remember, those three types of fibers are going to be slow oxidative, fast oxidative glycolytic, and fast glycolytic fibers. Alright, so now for the review questions. Why do the different types of fibers vary in contraction speed? So we talked about this at the beginning of the video. So go ahead and think about that. If you need to go back to the beginning of the video to understand that again, go ahead and do that. But hopefully you can remember that answer. And then another review question is, why do fibers have different appearances according to type? So why do some of them appear red and why do some of them appear white? So think about that and the reasons behind the different appearances. And if you understand those, I would say that you're in a fairly good position, that you've been able to understand the concepts of this video. But if you have any questions still, or you've had some questions come up after watching this that you want to know more about, feel free to reach out to us. We want you to do as well as possible on any tests that you might have coming up. But if you don't have any questions, have a great rest of your day and good luck with your studies. Hey y'all, it's Lance, and today we are going to be talking about degrees of muscle contraction. So go ahead and look at the outline if you want to, but we're just going to go ahead and jump right into things so you know exactly what we're going to be looking at for this video. Alright, so first off, before we talk about summation, there's a couple of important things that we want to touch on just to kind of help us know what summation actually is. So if you think about it, the twitch of a muscle fiber is always blank. And so this blank is going to be regarding the size of the twitch. And so as we know, for one muscle fiber, that twitch is always going to be the exact same size. So it's going to be the same size right here, alrighty. And then another question to pose right here is the force of a contraction is blank via blank. And so the force of a contraction, does that contraction always stay the exact same force? Does the force vary? And if so, how does that happen? And so you guessed it, the force of a contraction isn't always the same. And so the force of a contraction is going to vary. And that is actually because of summation itself. So let's go ahead and write that in. So the force of a contraction will vary because of summation. Alrighty, and we're going to have two different types of summation. We're going to have this first one down here. This is going to be a wave summation. This is also known as temporal summation, so wave summation. Alrighty, and then the second one down here, this is going to deal with motor units, and so this is going to be known as motor unit summation. Alrighty, so let's go ahead and talk about each one of these so we can better understand each one. and how they're similar and how they're different from each other. All right, so first off, let's go ahead and talk about wave or temporal summation. And so this type of summation is going to be generally associated with action potentials. And so kind of like a stacking of action potentials, you might say, that's going to result in this wave summation because there's going to be a greater force due to more action potentials happening. Alrighty, so this right here, this is going to be talking about how wave summation actually happens. And so we know that contraction actually happens when muscle fibers in a motor unit are depolarized. And so when these kind of stack on each other, think about if this is going to happen during the latent phase then or the relaxation phase. And so go ahead and pause the video, think about that if you need to. But this is actually going to happen during the relaxation phase. Alrighty, and so let's go ahead and just look at this down here, and then we're going to go ahead and talk about more of like how this actually happens to help it make sense for you. And so right here we have the force of a contraction from one muscle fiber, and so we have that same thing once again. And so this is two different action potentials in that same muscle fiber. But when this wave summation happens, when that depolarization happens again during the relaxation phase, it's going to result in something looking like this with more force behind it because that contraction is happening during the relaxation phase once again. And so those kind of stack on top of one another. So let's go ahead and talk about why exactly this happens. And so this thought question over here is how do subsequent contractions stack leading to summation? And so looking at this picture, think about how this might happen. Go ahead and pause the video, and then we're going to go ahead and answer this question. All right, so now that you've had some time to do that, let's go ahead and answer this. And so the answer is actually going to be with voltage-gated calcium channels. So voltage-gated calcium 2 plus. Alrighty. When you think about an action potential reaching the muscle fiber, we know that we're going to have this acetylcholine, we're going to have these sodium ions that are going to rush into the muscle fiber itself, causing the muscle fibers to be depolarized. That depolarization is going to reach this portion right here. And this, as we know, is going to be that sarcoplasmic reticulum. And then we can see these guys down here. And as we remember, those are going to be our voltage-gated calcium channels. And so these guys are going to play a really big role in this wave summation. And so we can see right here that the calcium ions have been released into the sarcoplasm during the contraction phase, but they're going to start coming back up, going into the sarcoplasmic reticulum again during that relaxation phase as well. When another action potential is going to come down, these voltage-gated calcium channels are going to open up again, and these guys are going to come back down into that sarcoplasm. And as we know, those are actually going to bind onto the C sites on troponin, which will then release actin and tropomyosin. And so when that actin is then released, we know that the myosin heads, those ATPases, they can then form that cross bridge with actin, leading to that muscle contraction actually happening again. And so that's kind of how that wave summation happens. There's just more calcium in this sarcoplasm right down here. So let's just go ahead and write that in. So this is the sarcoplasm. And so when these extra calcium ions come down into the sarcoplasm, we know that there's going to be more of the C sites that are being bound by calcium, which will then release more actin. And those actin binding sites that are then free can then interact with the myosin heads, and that's going to result in a cross bridge formation and a greater force for contraction because there's going to be more of those actin binding sites available and therefore more of a force behind that contraction. And so that's the basic gist of wave summation, and those are going to be associated with action potentials leading to a depolarization during the relaxation phase. So an example of wave summation is going to be with tetanus. And so here we can see that there's a lot of wave summation going on, but there's eventually going to reach a point right here where we're at the maximum tension. And so when maximum tension happens with these contractions, that's actually when we are in tetanus. And so let's go ahead and think about what the physiological basis of tetanus actually is. And so we know that there's going to be action potentials, and as we can see, there's going to be quite a few action potentials happening right on top of one another. And so we can see one here, 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 etc. And once we reach up here, we've had so many action potentials stacking on top of each other, there's so much of a wave summation happening that we reach that maximum tension. And so at this point in time, there's like tons of action potentials just kind of firing one after another. It's very continuous, and that is going to be what leads to tetanus. But think about what happens to those calcium channels when that happens. And so if there's a constant depolarization, all of the voltage-gated calcium channels are going to be open. So let's go ahead and write this in. So they're going to be open right here. And then that isn't really great because there's not going to be any calcium staying in that sarcoplasmic reticulum. And so we're going to just have all this calcium in the sarcoplasm, And so there's going to be a ton of these actin binding sites available for myosin to bind to, which will lead to this increased tension as well because there's going to be a lot of contraction going on. As we know, there's going to be really, really high ATP usage because we know that the myosin heads are ATPases. And so as those cross bridges are forming, those ATPases are doing their thing, and that's going to result in a lot of ATP usage. But additionally, we're going to have the active transport of calcium going back into the sarcoplasmic reticulum happening from that sarcoplasm. And when that happens, the voltage gated calcium channels are still open, and so it's kind of just a waste of ATP because the calcium is being pumped back in, but then it just gets released right back out because the voltage gated calcium channels are actually just open. And so the end result of this is going to be tetanus, or just that really rigid contraction, and that you can't really stop it because of the action potential just going. But eventually we're going to use so much ATP that we just kind of get fatigued, and so those contractions will actually just stop. And so tetanus will eventually end once that ATP usage has just fatigued us. And so that's going to be the end of tetanus. And so that's going to be a little overview of tetanus and like a really extreme example of wave summation, but it kind of gets that point across because we can see how it just builds upon each other until it reaches this maximum tension right there. All right, so let's go ahead and talk about the next type of summation, or motor unit summation. And so motor unit summation is kind of similar to wave summation based on the fact that things are going to stack on top of each other, but that's kind of where the similarities end. All right, so as we can see right here, this is kind of representing the tension from one motor unit. And so we have a small motor unit with just a little bit of tension, and they're going to have a larger motor unit with quite a bit more tension. And so right here we have this imaginary muscle, you can say. And so we can see that this is going to have 3 small motor units and 2 large motor units. And so we can see right here that we're going to have that tension from the 3 small, and then the tension from the 2 large. And during motor unit summation, the smaller ones are going to actually be recruited first, the smaller motor units, and then the larger motor units will be recruited. And so we're going to have this one recruited first, then this, and then the large ones will be recruited. And eventually the force of all those motor units will kind of stack on top of each other, leading to a larger force of that contraction. And so motor unit summation is basically using more and more motor units to lead to a greater amount of force behind the contractions. All right, so let's go ahead and think about this thought question right here. And so why is this summation beneficial? We know that in a muscle we're going to have a lot more motor units than this. And so when these guys all kind of stack on top of each other, that's just a small fraction of the motor units that are actually in the muscle itself. And so all 5 of these might be excited, and then we might have another 5 that are excited later on. And so basically the answer to this question is that it gives the muscles time to kind of relax and then kind of reset themselves so that they can be ready for the next motor unit summation that occurs when these motor units are then recruited for another contraction. So that's kind of why this is beneficial, just because we're not going to really get fatigued because we have a lot of motor units that we can fall back on to lead to this summation. All right, so let's go ahead and talk about a certain principle that is related to motor unit summation, known as the size principle of motor unit recruitment. And so there's going to be a specific order that motor units are recruited. And if you remember from that last slide, we're actually going to have the small motor units. Let's go ahead and write this in. So small motor units will be recruited first. So write that in right here. So small motor units. And then we're going to have our larger motor units recruited next. And so that's going to be what's next. So large. And as we can see, small motor units are going to be excited by the more excitable neurons, while large motor units are going to be excited by the least excitable neurons. And as you remember, small motor units are going to be those more precise movements, and so it's going to be going first, and then the large motor units are going to kind of step in, kind of like take the wheel, and those are going to lead to the force that we need to kind of make those muscles do exactly what we want them to do. And so just remember that the small motor units are going to be recruited first to kind of start the movement out, and then the large motor units are going to step in by the less excitable neurons, and those will lead to the exact kind of movement that we want to have for a specific contraction. And so that's going to be the size principle of motor unit recruitment, that the small motor units are recruited first, and then large motor units are recruited second. All right, so let's go ahead and talk about muscle tone now. So muscle tone is kind of a really important concept because when we're sitting here, like I'm sitting here right now, I don't really have to think about keeping my neck upright. I don't really have to think about sitting upright. It just kind of happens, right? And that's actually going to be due to muscle tone itself. And so muscle tone is actually going to be happening because of involuntary twitches. Like I said, we don't have to really think about doing this, so it's going to be involuntary. And these are going to be happening with very small groups of motor units. So this might include some small motor units, some large motor units, but it's going to be very small groups of motor units. All right. And we know that this is happening constantly throughout our body because we don't ever have to think about it. Our head just stays up. And so these are actually going to be happening due to continuous action potentials. So continuous right here. All right, and so the way that muscle tone happens is actually really, really beneficial. And so because there are small groups of motor units, there's not going to be like continuous contraction of a specific motor unit. So we might have one on the left side of our sternocleidomastoid, and then we might have one in the middle that gets innervated afterwards, and then one on the right. And then it might kind of just cycle through all of those different small groups of motor units. But because it's going to be different groups of motor units one after another, that gives those motor units that were excited time to relax. And so it's not going to really stack on top of each other with that wave summation. And so those muscles have time to relax. And so we're not really going to be fatigued at all with this. And so muscle tone is really cool how it works because it's just like these small motor units are being innervated and leading to these involuntary twitches, which just kind of help us be the way we are. It's not just like a pile of muscle and bone. And so this muscle tone is actually really, really important to kind of understand because it's how we're doing what we're doing right now. All right, and I just answered this thought question. So why does resting muscle tone not result in fatigue? And that is going to be because we have different motor units being recruited at different times. So different motor units at different times. Alright, so now let's go ahead and think about this. So do we have muscle tone in skeletal muscle, smooth muscle, and cardiac muscle? So no, actually. So we know that we're going to have it in skeletal muscle because that's how we're staying upright. And so we're going to have that for sure in our skeletal muscle. And then let's think about, do we have that in smooth muscle? So smooth muscle, we have like our esophagus, our digestive tract. That's going to have a lot of smooth muscle in it. And so this is actually going to have some muscle tone as well because it's going to help with our digestion. But cardiac muscle, this is the odd man out. This is actually one that does not have muscle tone. So it's kind of interesting that these two do. And a little way to remember this, they both have S in there. And so the two S muscles, those will have muscle tone, but then the odd man out there, the cardiac, that is not going to have that muscle tone. Alright, so let's go ahead and talk about some problems with muscle tone now. Hypotonicity and hypertonicity. And so hypotonicity is basically we're going to have less muscle tone than we should, and then hypertonicity is going to be we have more muscle tone than we should. Alrighty, so these are going to occur as a result to damage to the nervous system. And so hypotonicity is actually going to be a result of damage to the peripheral nervous system, so the PNS. And it can also result from low electrolytes. So low electrolytes right there. Alrighty, and so when this happens, the muscles are going to feel very flaccid, and so they're just not going to have that tone that we normally would expect from them. So they're going to feel flaccid. Alrighty, and so an example of this could be with like carpal tunnel syndrome. When that nerve is compressed in the carpal region, that can lead to this like flaccid feeling in our hand, and so this is going to be an example is carpal tunnel syndrome. Alrighty. So now let's go ahead and talk about hypertonicity. And so this is going to occur as a result to damage to the central nervous system. So an example of when this might happen is with like a stroke or something leading to this hypertonicity. And so when this happens, the muscles are going to feel very spastic and rigid. And so let's go ahead and write that in. So they're going to feel spastic and rigid. And this makes sense because we're going to have more muscle tone than we're used to. And so we're going to have more of a rigid feel in those muscles. And like I said, an example of this would happen with a stroke. And so that's just a quick little review of problems with muscle tone, this hypotonicity and this hypertonicity. Hypotonicity is when we have less tone than we should, and hypertonicity is when we have more tone than we should. Alrighty, so let's go ahead and just review this real fast if you need to. This is what we talked about in this video, this wave summation, motor unit summation, and then muscle tone. And then I have a few review questions that I want you to think about. So what are the differences between wave summation and motor unit summation? So the main similarity between them is that they kind of stack on top of each other, leading to a greater force of contraction. But I want you to think about what the differences are. If you need to go back and watch some of the video to better understand this, I recommend doing that. And then the next one is what is tetanus? And so this is going to be an example of wave summation. So think about how tetanus happens, the physiology behind it and all that. And so that's going to be this next question. So what is tetanus? The question after that, what size of motor unit is recruited first? So that's going to be with the size principle of motor unit recruitment. Hopefully you can remember which one's recruited first. If not, there's the answer in the video for you. And then this last one is explain the concept of muscle tone. This is a really important concept as it might show up on test day for you. And so I would recommend teaching this to a friend, a roommate, family member, whoever you have near you, because as you teach, you're going to be able to better learn it. And who knows, maybe you'll interest another person in learning more about degrees of muscle contraction. You never know. But hopefully we were able to answer any questions that you might have had during this video about degrees of muscle contraction. But if you had any questions come up during the video or we didn't answer all the questions you might have had, please feel free to reach out. We want to help you do as well as possible on the upcoming exams you might have. But if you don't have any questions, have a great rest of your day and good luck with your studies. Hey y'all, it's Lance, and today we're going to be going over how muscles work together to create movement. So this is kind of tying the entire muscular system together. So if you want to go ahead and pause the video, look at what we're going to be looking at today, you can, but we're just going to go ahead and jump right into it so we know exactly what we're going to be looking at today. Alright, so let's go ahead and talk about invertebrate movement. So this is going to be with invertebrates themselves as well as with unicellular organisms. So as you know, these organisms are going to have different types of movement than vertebrates would. And so with protozoans, these guys are going to be using cilia and flagella. And so these would be examples of protozoans. So we can see that we have flagella and we have cilia, and those are on the outside of the organisms. And so those are going to help the organisms actually move around. And so those are going to be with protozoans. Then we're also going to have amoebas. And amoebas are going to use pseudopods to help themselves move around. And so we can see these pseudopods extending out, and this is going to be how those amoebas move around. And so like this is very different from how we as vertebrates move around. So it's kind of interesting to look at what other organisms do to move compared to us. And then we're also going to have something called a hydrostatic skeleton, and these are with invertebrates such as nematoda. And they're going to use this hydrostatic skeleton to help them move around. And so we can see that there's going to be movement towards that head end that we see right here. And so that's going to be with a hydrostatic skeleton. We're going to talk more about this in the biodiversity chapter, so don't worry too much about these right now. But I just wanted to make you guys aware that we have these different types of movement depending on what type of organism we are. So we're going to have cilia, we're going to have pseudopods, flagella, and hydrostatic skeletons, and those will help these invertebrate organisms move around. All right, so let's go ahead and talk about vertebrate movement now. And so I want you to try to think about what the two main types of muscular contraction is. So if you need to go and pause the video to think about this for a little bit, feel free to do that. But if you feel pretty certain, just go ahead and write these in right here. We're going to have two types, and we're going to go ahead and write these in right now. So first off, let's talk about this one right here. We're going to have isotonic contractions right here. So let's go ahead and write that in, isotonic. All right. And then we're also going to have these ones over here, and these are going to be isometric. So they're both going to have iso in the name, but one's going to be isotonic and one's going to be isometric. So let's go ahead and write this one in. All right, sweet. So as we can see, there's just one type of isometric contraction, but there's going to be two different types of isotonic contractions. So if you remember these, go ahead and fill them in with me. But if not, we're going to go ahead and learn this together. All right, and this first one that we're going to talk about, these are going to be our concentric contractions. So let's go and write that in. So concentric. And this one down here, this is going to be an eccentric contraction. So let's go ahead and write that one in as well. So eccentric. Alrighty, so let's go ahead and dive into these so we can know exactly how each of these differ from each other and exactly what they are. Alright, so let's go on and talk about these isotonic contractions. So we have a little bit of a thought question for you right here. So I want you to go ahead and pause this video and see if you can fill in these two blanks. So what does iso mean and what does tonic mean? Alright, so now that you've had some time to do that, let's go ahead and fill these in. Alright, so as we know, we're going to have iso, and iso, as we know, is going to mean the same. So same right here. And then we're going to have tonic, and tonic can mean a lot of different things in different scenarios. But in the sense of isotonic contractions, this is going to mean tension. So tension is going to be right here. All right. So right here, we're going to have isotonic. So this means the same tension across the entire contraction. So let's go ahead and think about this question now. Why is curling a dumbbell an isotonic contraction? So if you think about it, the dumbbell is going to stay the exact same weight the entire time we're curling it. And so that tension is going to stay the same throughout the entire thing. And so this type of movement with curling a dumbbell, that's always going to be isotonic just because that tension is going to remain the same throughout the entire contraction. So this is going to be the same tension throughout the contraction. Alrighty. So as we know, there's going to be two different types of isotonic contractions. So let's go ahead and talk about those. We're going to have these concentric contractions, like I was saying, and then we're also going to have these eccentric contractions. So let's go ahead and talk about each one of these individually. So with concentric contractions, an example of this would be curling the dumbbell up. And so let's go ahead and draw an arrow in right here. So we can see it's going up like so. That's going to be a concentric contraction. And so as we can see, the muscle length is getting a little bit shorter because it's curling that in. So the muscle length is going to get shorter. Alrighty, and as we see, the bone it inserts on, so this is going to be our biceps brachii, and so we know that this is going to insert on the radius, and then it's going to originate from the humerus, and so this is going to be pulling that radius closer to the humerus, and so the bone it inserts on gets closer to the bone that it originates from. So let's write that in, so closer. Alright, and now let's go ahead and talk about these eccentric contractions. And so with eccentric contractions, it's going to be the exact opposite, so it's kind of convenient for us. And so the muscle is actually going to lengthen a little bit, so it gets a little bit longer. Alright, and the bone it inserts on is actually going to get farther away from the bone that it originates from. And so it's going to be farther. Alrighty, and so that's going to be this type of movement going down away from that. So right here, we're going to have concentric contractions, and then down here, going away from that bone that it originates from, that's going to be our eccentric contraction. All right, so a little mnemonic to help you guys remember this is going to be with the muscle length and like where the bone positions are. So with concentric, we're going to have the bone that the muscle inserts on get closer to the bone that it originates from. So that's going to be a C. So that's going to be a C to a C. So concentric is going to get closer. And then with eccentric, the muscle is actually going to get longer. So you can say that the muscle is going to elongate. And so that's going to be an E going to an E right there. So if you ever get in a bind, you can't quite remember this on test day, you can remember C is going to be with that C, so concentric is going to be closer. And then we're going to have E with E, so eccentric is going to elongate that muscle. So hopefully this will help you on test day, but if not, it's just a fun little fact to help you remember it down the road. Alrighty, so let's go ahead and talk about the isometric contractions now. Alright, so as we know from the previous thought question, we're going to have iso, which means the same. So let's just go ahead and write that in real fast. So iso means the same. And now I want you to pause this video, think about what does metric mean. Alright, so now that you've had some time to think about that, metric, as we know, is going to be with the length. So right here is the length. So isometric contractions are going to be the same length even though the muscle is contracting. So we can see that in an example right here. And so this guy right here is just holding that dumbbell straight out in front of him. That elbow's at that 90-degree angle. And so we can see right here before that dumbbell's being held, we have this length of the biceps brachii. And then right here, after that dumbbell has been grabbed and he's holding it out, we can see that it kind of contracts right here, but that muscle length is actually going to stay the same. It's not going to shorten or elongate. And so that kind of answers the question, why is holding a dumbbell an example of an isometric contraction? And so the muscle is still contracting. So let's go ahead and just write this in. So muscle contracts, but the length stays the same. Alright, so that's going to be an example of an isometric contraction. So this is a quick recap of all 4 different types of contractions. We're going to have isotonic contractions, which are going to be the same tension throughout the entire contraction. And in that type of contraction, we're going to have 2 different types. We're going to have concentric contraction, which means the bone the muscle inserts on is going to get closer to the bone that it originates on. And then we're going to have eccentric, which is the exact opposite. That muscle is going to elongate. And then we're going to have these isometric contractions right here. And that is when the muscle contracts, but the length is going to stay the same throughout the entire contraction. Alrighty, so just one more thing we want to talk about. Let's go ahead and talk about functional groups of skeletal muscles. And so these are going to be agonists, antagonists, and synergists. So first off, let's go ahead and talk about agonists. So this is going to be this one right here. So agonists are going to be the muscles that are involved in performing the desired movement. So basically, this is going to be the muscle that performs the movement we want. So perform the movement. Alrighty. And when I think about antagonists, I think about like in like a book, we're going to have like our main characters and they're going to have our antagonists. So our protagonist and antagonist, that's what the word is. And so with antagonists, that's going to be like the opposite. And so this is going to be the muscle that performs the opposite movement. All right, so let's go ahead and write that in. So performs opposite movement. All right, so now let's go ahead and talk about synergists. So synergists are basically going to be muscles that help stabilize the joint that the action is happening across, as well as just helping that agonist do its thing. So let's go ahead and write that in. So basically they're just like a little helper for the agonists. So help the agonists or the primary movers. Agonists are also known as these primary movers. So let's go ahead and write that in on agonists as well, just to help remind us. So also primary movers. Alrighty, sweet. So as we can see down here, we have a picture of some muscles of the upper arm. And so these muscles that we can see, we're going to have the biceps brachii, we're going to have triceps brachii, and we're going to have the brachialis. And so each of these muscles is going to be either an agonist, an antagonist, or a synergist. And so the action that we're going to want to do is going to be to flex that elbow. And so go ahead and think about what the agonist for this muscle would be. And so the main or the primary mover, that's going to be our biceps brachii, so we're just going to go ahead and outline that. So biceps brachii is that agonist. And as we know, the opposite movement is going to be done by the triceps brachii, and so that's going to be our antagonist. Let's go ahead and outline that as well. So antagonist is going to be the triceps brachii. And then our synergist, where you can see that we have this muscle right here, the brachialis, and that muscle is going to be helping the biceps brachii. And so this one right there, that brachialis, that's going to be known as a synergist. Alrighty, so those are going to be examples of functional groups of skeletal muscles. So once again, we just have those agonists or those primary movers doing the actual movement, the antagonists performing the opposite movement, and the synergists, which just help the agonists. Alrighty, so let's go ahead and talk about this review side right here. So we just talked about all of these concepts, but I also have a couple of review questions for you. And so one of those review questions is, what are the main differences between isotonic and isometric contractions? So go ahead and think about the definitions for each one of those, about what's going to stay the same, what's going to be different. And so go ahead and think about that. And then I want you to go ahead and give an example of an agonist, an antagonist, and a synergist in the leg. And so we talk about those with the muscles of the upper arm, but there's also going to be examples in the leg. So I want you to go ahead and think about that and explain it to a roommate, a friend, whoever you want to. But you'll be able to learn better by teaching it to someone else. So I'd recommend doing that. All right, that's going to be it for this review. But if you do have any questions, please feel free to reach out. We want you to do as well as possible on test day. So if there's like a concept you didn't quite understand, or maybe you had a question come up during the video that I didn't answer, go ahead and reach out to us. We want to help you do as well as possible, like I said. But if you don't have any questions, have a great rest of your day and good luck with your studies.